Hello, my dear students. Welcome to my today's online class, that is continuation of the chapter Fertilization. In my previous videos, I have taught you the definition, types, mechanism of fertilization, and significance of fertilization. Now we have to understand one more phenomenon or concept, that is polyspermy. Polyspermy, pathological and physiological polyspermy with suitable examples. And then you have to understand the mechanisms to block polyspermy in monospermic forms. There are two types: fast block mechanism and slow block mechanism. So if we are able to have a discourse on these topics, we are done with our duty today. Okay, now let us discuss about this. So this is me, and you can see the diagram here. You can see the egg here, and many sperms trying to make an entry into the egg. There are around 6 sperms here, but you can make out that only one sperm has managed to get into the egg and managed to fertilize it by bringing about amphimixis, whereas the other 5 sperms are simply present, being attached to the plasma membrane, right? So this depicts the typical polyspermy. Okay, now so in biology, what do we mean by polyspermy? It describes an egg that has been fertilized by more than one sperm. Monospermy, mono means single, sperm means sperm. Monospermy, where the egg being fertilized by a single sperm. And contrary to this, polyspermy refers to, poly means many, many sperms, fertilizing one ovum. Okay. Then diploid organisms. Diploid means 2n number of chromosomes. Normally contain 2 copies of each chromosome, all of us know, which are obtained one from each parent, one from maternal parent and the other one from paternal parent, right? Then, so the cell resulting from polyspermy, on the other hand, contains 3 or more copies of each chromosome, because the ovum would have received the chromosomal complements from many sperms, right? So depending upon how many sperms have made an entry, that many number of sets of chromosomes you find. One derived from the egg and one each from multiple sperm. So such type of fertilization often results in an unviable zygote, because normal diploidy is replaced by triploidy, tetraploidy, etc. Right? Now, this may occur because the sperms are too efficient at reaching and fertilizing the eggs due to the selective pressures of sperm competition. Okay? So this is a sort of mad race of sperms to reach the egg and ultimately fertilize. In National Geographic channel, normally one episode only comes. You can just watch it. So the title only is this, Mad Race of Sperms. Okay? You can just watch it to have a better understanding about this. Then, such a situation is often deleterious to the female, because the male-male competition among the sperm spills over to create sexual conflict, and as a result, you will find more than diploidy condition in the fertilized zygote, which is not viable, which ultimately results in an unviable zygote, and whole purpose of reproduction is going to be a failure. Okay. Now, we shall talk about the types of polyspermy. One is physiological polyspermy, where polyspermy by nature is practiced in the animals, and the other one is pathological polyspermy, which is seen in animals which follow monospermy. Okay. So let us understand the types now. First we shall study the pathological polyspermy. This happens when the egg normally accepts more than one sperm. But even though there is entry of multiple sperms, with which will fuse their nucleus with the nucleus of the egg. Okay. So normally accepts more than one sperm. Physiological polyspermy happens when the egg normally accepts more than one sperm. But please note, only one of the multiple sperms will fuse with its nucleus with the nucleus of egg. Though many sperms manage to enter the egg, all sperms will not contribute their male pronucleus to fertilize the egg. Out of that many number of sperms which make an entry, ultimately only one sperm will fuse its nucleus with the nucleus of the egg. Okay. Now, so this physiological polyspermy is present in many species of vertebrates and invertebrates. Some species very cleverly utilize this physiological polyspermy as the proper mechanism for the developing their offspring. Okay. So they take physiological polyspermy to their advantage. One sperm will fertilize the ovum and the others will help in nursing function. Okay. So proper development of the embryo takes place because of the contribution from other sperms. Okay. Now, for example, birds, ctenophores, reptiles, amphibians, in all these cases, you can see physiological polyspermy. Okay. Now, there are different points to be noted here. Let me discuss one by one. Urodele amphibians, that means tailed amphibians, okay, salamanders, etc., newts and salamanders, and then cartilaginous fishes, birds and reptiles. They undergo physiological polyspermy because of the internal fertilization of the yolky eggs. Okay. Then sperm triggers egg activation by the induction of a free calcium ion concentration in the cytoplasm of the egg. Right? This I have already taught you. So the sperm triggers egg activation how? By inducing the increase in the concentration of calcium ion levels in the ooplasm. Cytoplasm of the egg is called ooplasm. Okay. Then this induction plays a very critical role in both the physiological polyspermy and monomeric polyspermy species. Okay. Then the rise in calcium causes activation of the egg. The egg will then be altered on both the biochemical and morphological levels. Okay. Then in mammals as well as in sea urchins, echinoderms, the sudden rise in calcium concentration occurs because of the influx of calcium ions within the egg. Okay. Then these calcium ions are responsible for cortical granule reaction, I'll come to it later, and are also stored in the egg endoplasmic reticulum. Calcium ions are stored in endoplasmic reticulum of the egg. Okay. Then, so unlike physiological polyspermy, monospermic fertilization deals with the analysis of egg calcium waves, as this is the typical reproduction process in all species. Alright. Then, species that undergo physiological polyspermy have polyploidy preventing mechanisms that act inside the egg. Then, this is quite different from normal polyspermy block on the outside of the egg. So, different groups of animals have devised their own ways and means, ultimately to see to it that the act of fertilization is a big success, and therefore perpetuation of the race on this Mother Earth is also a big success, right? Then, so the eggs of zebra finches and chickens, they require multiple sperm, not 1, 2, 3 or 4, from 10 to hundreds of sperms to penetrate the egg and to ensure successful fertilization and growth of the bird embryo. Okay. Now, how are you going to block this polyspermy? Polyspermy is very rare in human reproduction, and the decline in the number of sperm that swim to the oviduct is one of the two ways that prevents polyspermy in humans. Okay. Then, the other mechanism is blocking of sperm in the fertilized egg. So, if the egg becomes fertilized by multiple sperm, the embryo will then gain various parental centrioles and whole thing is going to be a chaos. Now, so when this happens, there is a struggle for extra chromosomes. So this competition causes disarrayment in cleavage furrow formation, and therefore the normal consequence is the death of the zygote, right? So only 2 cases of human polyspermy leading to birth of children have been reported to date. Now, after knowing about this physiological polyspermy, it is the time to understand the mechanisms that have been devised by the organisms to prevent polyspermy. One is fast block of polyspermy and the other one is slow block of polyspermy. Both I am going to teach now. So the eggs of sexually reproducing organisms, they are adapted to avoid this situation. That is the beauty of the nature. Always it provides congenial conditions for anything to function in an optimum way. And the defenses are particularly well characterized in sea urchin, which respond to the acceptance of one sperm by inhibiting the successful penetration of the egg by subsequent sperms. And similar defenses exist in other eukaryotes also. Now, so the prevention of polyspermy in sea urchins, depends on the change in the electrical charge, depends on the change in the electrical charge across the surface of the egg, which is caused by fusion of the first sperm with the egg. Carefully listen. So, fast block mechanism involves change in the electrical change across the surface of the egg, which is caused with the first entry of the first sperm within the egg. Okay. So, unfertilized sea urchin eggs, they have a negative charge inside. Okay. But once fertilization takes place, the charge becomes positive, right? So, there is change in negative charge to positive charge with fertilization. Alright. When sea urchin sperm encounter an egg with a positive charge, in the next PPT I will show you the diagram, sperm egg fusion is blocked. Alright. So, thus after the first sperm contacts the egg and causes the change, subsequent sperms are prevented from fusing. This is because sperms have positive charge on their membranes, and when the inside of the egg also has positive charge, positive, there is repulsion. This is what exactly happens. Therefore, there is no scope for the second sperm to get into the egg inside. Alright. So, this electrical polyspermy block is thought to result because of a positively charged molecule in the sperm surface membrane being repelled by the positive charge at the egg surface. Okay. Now, let's see here. So, electrical polyspermy blocks operate in many animal species including frogs, clams, that is mollusks, and marine worms, but not in several mammals that have been studied like hamster, rabbit, mouse, etc. In the diagram you can see, so in the first diagram, there is negative charge, right, inside the ovum, and this first sperm is approaching. In the second one, the sperm has approached, and in the third one, the sperm surface has touched the egg surface. With this, there is sudden shift from negativity to positivity, and the first sperm makes an entry inside the egg, and in the fourth one, the second sperm is being avoided because of positive charge. This is what I exactly meant by electrical polyspermy block, that is fast block mechanism. Okay. Then, in species without an electrical block, polyspermy is usually prevented by secretion of materials that establish a mechanical barrier to polyspermy, right? So, this is all what I wanted to tell you about electrical polyspermy block or fast block mechanism to prevent polyspermy in animals. Okay. Then, so now, animals such as sea urchin, this is something fantastic you should know. They have a two-step polyspermy prevention strategy. Very nice, very clever. With the fast but transient electrical shock, suspended electrical block suspended after the first minute or so by a more slowly developing permanent mechanical block. What I meant to say by this statement is, see, till the first minute after the first sperm enters the egg, so this electrical polyspermy block or fast block mechanism operates. Then, please note, this is a temporary phase and this can happen only for 1 minute. So, what should do after the first minute? So, the electrical block goes off. Okay. Later, the sea urchin eggs, they adapt towards developing permanent mechanical block, that is slow block mechanism or cortical reaction. Till cortical reaction is activated, the polyspermy is avoided by fast block mechanism or electrical block. Okay. When once that is over by first minute, then from second minute or so, slowly slow block mechanisms start operating. Okay. That is cortical reaction. So thus sea urchins have developed a two-step strategy to begin with fast block polyspermy, fast block mechanism, then that is followed by slow block mechanism. So electrical polyspermy prevention is followed by cortical reaction, right? This is what I meant by that statement. So electrical blocks are helpful in species where a fast block to polyspermy is needed. due to the presence of many sperm arriving simultaneously at the egg surface, as occurs in animals such as sea urchins, where fertilization is external and simply millions of sperms are dumped into the area where the spawn is present. So here, electrical fast block mechanism is needed. All right. Then, so in sea urchins, fertilization occurs externally in ocean such that hundreds of sperm encounter the egg within several seconds. Therefore, as a first step, they follow electrical block, right? And as a second step, they follow cortical reaction block. Now, we shall talk about the slow block of polyspermy. In other words, cortical reaction. In brief, I have referred this while discussing about the mechanism of fertilization. Now we shall discuss the same in detail. In mammals, where the fertilization is internal, to begin with only, during copulation, fewer sperm reach the fertilization site in the oviduct in other animals and fallopian tube in humans. So this may be the result of female genital tract being adapted to minimize the number of sperm reaching the egg. Nevertheless, polyspermy preventing mechanisms are essential here also, but a different strategy is followed, cortical reaction, which modifies the extracellular coat of the egg, okay, or the zona pellucida as you see in mammals, and additional mechanisms that are not well understood that modify the egg plasma membrane. All right. So the zona pellucida, by now you must be knowing, this is a layer, okay, after the plasma membrane, what you see in mammals, zona pellucida, then comes zona radiata. All right. The zona pellucida is modified by serine proteases that are released from the cortical granules. Okay. So this is called zona reaction. The reaction occurring in zona pellucida is called zona reaction. Reaction occurring in cortical region of the egg is called cortical reaction. Okay. Now, here you can see a plate showing you the cortical reaction. So here, what happens is, see, this causes, here you can see, granules from the cortex which are released through exocytosis, right? And enzymes released during this reaction harden the fertilization membrane, right? This is so slow block to polyspermy, all right? Here same thing in diagrammatic manner I have given, which you can write in your answer books. You can see here the egg membrane, white line membrane, peri-white line space, and these cortical granules, okay, slowly releasing their contents, and these forming a sort of lamellar structures and converting white line membrane into fertilization membrane and preventing the entry of sperms, more sperms, in a way preventing polyspermy. All right. Okay. So these two diagrams you have to understand. So the proteases destroy the protein link between the cell membrane and the white line membrane. Okay. Remove any receptors that are present on the membrane to which the multiple sperms can bind and help to form the fertilization envelope. Okay. So from the cortical granules. The cortical reaction occurs due to calcium oscillations inside the oocyte. I think this I have discussed much in detail when I was talking few minutes earlier, right? Then, so what triggers such oscillations is PLC zeta. What it is? PLC zeta. It is a phospholipase enzyme. Okay, phospholipase enzyme, very unique to sperm, and that is very sensitive to calcium concentrations. Then, when the first sperm gets inside the oocyte, it brings in PLC zeta, which is activated by oocyte's basal calcium concentrations. This initiates the formation of IP3 and causes calcium release from endoplasmic reticulum stores. I have already told you, calcium ions are present in large quantities in endoplasmic reticulum. So this generates oscillations in calcium concentration that will activate the oocyte and block the polyspermy. This may appear little complicated. I don't see any complication, but still. So if you study once or twice, listen to the video and study the material what I am going to supply to you, I think the concept should be clear. Here I am talking about triggering of oscillations, which is because of PLC zeta. It is a phospholipase, very unique enzyme present in sperms, and this is very sensitive to calcium concentration. Okay. The moment the first sperm enters the oocyte, it brings with it PLC zeta, and it is activated by basal calcium concentrations, and this initiates the formation of IP3 and causes calcium release from endoplasmic reticulum stores, thus generating the oscillations in calcium concentration that will activate the oocyte and block the polyspermy. So dear students, in this class I have discussed the polyspermy concept, monospermy concept, physiological polyspermy concept, and pathological polyspermy concept, and normally the mechanisms employed by animals to prevent polyspermy in monospermic conditions. Okay. So fast block mechanism and slow block mechanism. For further more details, you can always study the embryo developmental biology by Gilbert. Very nice book. He gives in detail these concepts. I am going to give you here few YouTube links where they have one video is pertaining to the cortical reaction and zonal reaction, and the other video is about electrical polyspermy block. After listening to my lecture, do watch these videos. Okay. Then so in this plate I have given the textbooks to refer and web source, right? Web sources. Then I have given around 20 questions. The question bank is here. All 1 mark question, 3 mark questions, 5 mark questions, 10 mark questions, around 20 questions. Sorry. You can take a blue book and start answering all these questions. Any moment I may give you unit test. Okay. So there are around 20 questions, right, in compliance with the evaluation guidelines from Bangalore City University. Okay. So now it is the time for me to thank you and close this class. See you next week in Teachment interactive class of these videos. Till then, stay blessed. Thank you.